Just like the genetically modified fruit, there are genetically modified organisms. An organism whose genetic material has been altered using genetic engineering techniques is termed as genetically modified organism. An organism that have been genetically modified include microorganisms such as bacteria, yeast, plants, fish, and mammals. When genes are inserted, they usually come from a different species, which is a form of horizontal gene transfer. In nature, this can occur when exogenous DNA penetrates the cell's membrane for any reason. To do this artificially may require attaching the genes to a virus or just physically inserting the extra DNA into the nucleus of the intended host with a very small syringe or with very small particles fired from a gene gun. Agrobacterium's ability to transfer genetic materials to plants or the ability of lentivirus to transfer genes to animal cells are natural examples of gene transfer. Bacteria were the first organism to be modified in the laboratory due to their simple genetics. These organisms are now used for several purposes and are particularly important in producing large amounts of pure human proteins for use in medicine. Genetically modified bacteria are used to produce the protein insulin to treat diabetes. Similar bacteria have been used to produce clotting factors to treat hemophilia and human growth hormone to treat various forms of dwarfism. The first transgenic animal was a super mouse created by Ralph Brinster and Richard Parmenter in 1982. It was created by inserting a human growth hormone gene in mouse genome. The offspring was much larger than the parents. Mouse is a common animal that is used for transgenic experiment. Other animals include pig, goat, cow, sheep, fish, etc. The production of transgenic animals involves the following methodology. The first step is the construction of a transgene, and the transgene is made up of three parts: a promoter sequence, the gene to be expressed, and the termination sequence. The second step is the introduction of the foreign gene into the animal through pronuclear microinjection method or by embryonic stem cell method. In microinjection method, a female animal is superovulated and eggs are collected. The eggs are fertilized in vitro. The transgene containing solution is injected with the male pronucleus using a micropipette, and eggs with the transgenes are kept overnight in an incubator to develop to a two-cell stage. The eggs are then implanted into the uterus of a pseudo-pregnant female, that is, the female which has been mated with a vasectomized male the previous night. The second method is the embryonic stem cell method. The transgenic animals can be created by manipulating embryonic stem cells. Embryonic stem cells are obtained from the inner cell mass of a blastocyst. Transgenes incorporated into the embryonic cell by microinjection, by a retrovirus, or by electroporation. Transgenic stem cells are grown in vitro and then they are inserted into a blastocyst and implanted into a host uterus to grow normally. Then as a screening procedure, the transgenic progenies are screened by PCR to examine the site of incorporation of the gene, and some transgenes may not be expressed if integrated into a transcriptionally inactive site. Further animal breeding is done to obtain maximal expression. Heterozygous offsprings are mated to form homozygous strains. Let's see some transgenic animals example. The first one is transgenic fish. In that, the super fish, it is having increased growth and size, and growth hormone gene are inserted into fertilized egg. Transgenic salmon grows about 10 to 11 times faster than the normal fish. Then is a glowfish. It's a genetically modified freshwater zebra fish produced by integrating a fluorescent protein gene from jellyfish into embryo of the fish. So such a glowy appearance will be there. Transgenic mouse are especially used for the experimental or study purpose. The first one is the Alzheimer's mouse. In the brain of Alzheimer's patients, dead nerve cells are entangled in a protein called amyloid, and mouse made by introducing the amyloid precursor gene into fertilized egg of mice. Then there's the onco mouse. It's a mouse model to study cancer. It's made by inserting activated oncogenes. Then there's a smart mouse. It's a biological model engineered to overexpress NR2B receptor in the synaptic pathway, and this makes the mice learn faster like juveniles throughout their lives. The next one is a transgenic pig. In that, the enviro pig, because these pigs have a trouble fully digesting a compound known as the phytate found in many cereal grains used to feed them. The transgenic pigs are created by introducing the phytase gene, which is taken from the bacteria E. coli. Phytase enzyme is produced in the salivary gland of pig, and it degrades indigestible phytate with the release of phosphate that is readily digested by the pigs. Then there are transgenic pigs that is developed for organ transplant. The pigs with human genes in order to decrease the chance of organ rejection by human body. Then there are transgenic cattle the transgenic cows are made to produce proteins, lactoferrin and lactoferrin in their milk. Also prion-free cows that is resistant to mad cow disease. Then there is transgenic sheep that is for good quality wool production, and there is transgenic goat that could express tissue plasminogen activator, antithrombin 3 and spider silk etc. in milk. Next is the transgenic rabbit alba, the enhanced green fluorescent protein bunny created in 2000 as a transgenic artwork. Then there is transgenic monkey Andy. It stands for inserted DNA when spelled backwards. An engineered virus was used to insert the harmless gene for green fluorescence protein into Andy's rhesus genome. Andy proves that transgenic primates can be created and can express a foreign gene delivered into their genome. So let's see the importance of transgenic animals. The medical importance include the disease model. They are used for research and study purpose, and as a bioreactors for pharmaceuticals, for the production of pharmaceutical products. And in xenotransplantation, xenotransplantation is the transplantation of living cells or tissues or organs from one species to another. And the agricultural importance include the disease-resistant animals for improving the quality and quantity of milk, meat, eggs, and wool production. And the industrial importance, the toxicity-sensitive transgenic animals to test chemicals, and the spider silk in milk of goat. These silk protein from the goat's milk are used for a variety of applications due to its strength and elasticity. It have medical uses such as for the making artificial ligaments, tendons, for eye sutures, for jaw repair. And coming to its industrial importance, these are also used in bulletproof vests and improved car airbags. Now let's see some of the common concerns that is associated with the genetic modification. First one is the allergenicity. GM foods have the potential to cause allergic reaction in general, and this risk is comparable to the risk associated with traditionally grown food. However, the proteins produced by newly introduced genes have the potential to cause an additional allergic response. To prevent such allergenicity, the transfer of genes from commonly allergenic food is discouraged unless it can be proven that the protein produced by the introduced gene will not be allergenic. Another potential risk is the introduction of an entirely new protein that did not previously exist in the food chain. The next one is the gene transfer. The transfer of genetic material from genetically modified food to the cells of the human body or the bacteria in the intestinal tract. DNA from ingested food is not completely degraded by digestion, and small fragments of DNA from genetically modified food have been found in different parts of the gastrointestinal tract. This could result in horizontal gene transfer due to absorption of DNA fragments by gut microflora or somatic cells lining the intestinal cells. Scientists, however, postulated that the uptake of genetically modified DNA into the cells of the gastrointestinal tract will not have any biological consequences because this DNA will be degraded in the cell. The next one is the increase in anti-nutrients. These are substances that interfere with the utilization of nutrients, and the insertion of a new gene may lead to an increase in the existing level of anti-nutrients. For example, glyphosate-resistant Roundup Ready soybean has been shown to increase anti-nutrients. In sheep and cattle, heat-stable anti-nutrients such as phytoestrogens, glycinins, phytic acid have been found to be cause infertility, allergic reactions, and decreased availability of phosphorus and zinc. The next concern is the use of viral DNA in plants. Most GM crops utilize the cauliflower mosaic virus 35S promoter to switch on the introduced gene. And there's a controversy as to whether the CMV35S would be horizontally transferred and cause disease via carcinogenesis, mutagenesis, reactivation of dormant viruses, or generation of new viruses. And let's see some of the pros of genetically modified food. The improved yield, more resistant to disease, less likely to be damaged by insect, tolerance to herbicides, better nutritional value, increased shelf life, improvement in health and environment, better climatic survival by increasing tolerance to drought, flood, or crusty conditions to high crop yields, reduced farm cost, increased farm profit. Some of the criticism of GM foods are, the first one is related to environmental hazard, is that the unintended harm to the other organisms. Also, reduced effectiveness of the pesticides, that is pesticide resistance develop. Then gene transfer to non-target species, that is there are chances that crossbreeding occur, and transfer of herbicide resistance from crops to weeds will be a major problem. The super weeds will then have herbicide tolerance as well. The human health risk includes the allergenicity as we discussed earlier and other unknown effects on human health. And the economic concerns, like it's a lengthy and costly process. And the big companies like Monsanto, Novartis can patent their product and make profit by exporting them, but the farmers from developing countries cannot afford it. So there will be more gap between the rich and the poor. And in case of transgenic animals, it's like blurring the lines between the species by creating transgenic combinations. There may be health risks associated with transgenics, and there may be long-term effects on the environment when transgenic animals are released into the field. And various bioethicists argue that it is wrong to create animals that would suffer as a result of genetic alteration.